Okay, let's see, where are we? We're talking about meiosis. And just to remind you some of the things. Generally speaking, when we talk about meiosis, we are talking about the conversion of a diploid cell into two haploid cells. And that generally means we're going to separate the different alleles for a gene from each other. Now on the surface, it looks pretty easy, but when you complicate things by having multiple genes and multiple chromosomes and multiple genes per each chromosome, things can get a little confusing. We should be able to look at a picture, at least a very simplistic picture, and think about what are all the possibilities, which will lead us directly into thinking about Punnett squares just a little bit. But if we assume that when we see this picture, what do we know about a cell when we see a genotype listed? Do we know how many chromosomes are there? No. How do you know that could be one chromosome? Could be two chromosomes. Because you don't know the relationship of A and B are to each other. So we have two sort of starting out points when we look at genotypes. And one possibility is that A and B are on different chromosomes, in which case we get this starting out. Or the possibility is they're on both the same chromosome. And here we have a little bit of a different possibility, because you don't know what is the relationship of A to B. And so just looking at genotypes tells us something, but doesn't tell us all the things we need to do. And depending on what things look like to start with, different processes of meiosis will come to bear to make changes. And so if you think about crossing over, which is the DNA exchange between homologous chromosomes, if the genes of interest are on different chromosomes, crossing over has no benefit or bearing whatsoever. It's only when we look at genes that are on the same chromosome that crossing over has the ability to create some new variations. Which is why I like to think about this as an intra-chromosomal change, because it only affects the things on one chromosome and not the things in a separate chromosome. But depending on where you start, that's going to affect how you would end. So something we also need to think about is if we don't know the relationship of the A to the B, we don't know what's going to happen if crossing over happens. So we're still limited with information, but as we go forward, we'll try to think about ways that we can approach looking at how do we determine what's going on here. But again, just to remind you, crossing over deals with changes within a single chromosome. We're going to try to reshuffle the traits that are encoded in a single chromosome number. Crossing over is a form of recombination, which is typically a rare event, so it's not always going to happen, especially when you have a really long chromosome, but it will happen at least once per chromosome, so there's always going to be some variability introduced to the process of crossing over. Anytime you start breaking a chromosome, you can have potential issues. Crossing over does require the DNA to be cut. On chromosome 1 of mom and chromosome 1 of dad at the same position, and DNA to be physically moved from one side to the other. We're moving double-stranded DNA from one location to a new location on the other chromosome. And cutting DNA can have some implications. These are what we call chromosomal abnormalities that are caused through recombination or crossing over errors. Some of them we talked a little bit about, like duplication and deletions, are things that we've heard about before. Does anybody remember in what context we heard about duplication and deletion of DNA? So polymorphism, the biggest polymorphism. And so the copy number variation is CNVs. So that's the thing we want to sort of put together as we go forward is some of these changes are a particular type of polymorphism. They're also going to be large scale changes, meaning it's not a single gene that's going to be duplicated, deleted, inverted, or translocated, but a big chunk of the chromosome. It's hard to say exactly how much, but it's not a single nucleotide. It's not a repeat of 3 or 4 every once in a while. It's a large chunk of DNA that gets moved. And sometimes that creates issues. So deletion and duplication is pretty straightforward, right? You lose some chunk of DNA, which typically means more than one gene has been lost, or gene and regulatory region or something else could also be lost. Same thing for duplication, but in the opposite context. A lot of students struggle with the idea of inversion. Can anybody try to explain why inversion might be bad? But first, maybe we need to talk about what inversion actually means. Let's do that first. Then I'll ask you to explain why it is actually bad. Then we have translocation. Sorry, I started getting ahead of myself. Let's not get ahead of myself. Now, typically, these things fall in the same category as aneuploid states. Remember aneuploid? Who remembers the two different types of aneuploid states? One's an M, one's a T. They both end in my. Yeah, so what's the thing about monosomy and trisomy is you have the wrong number of chromosomes, either one too few or one too many. And again, these are large changes that we're talking about. Even though you're not getting an entire chromosome worth of extra information or losing information, you're changing the context of what that information is. And so because of these large changes, a lot of times they act and the cell responds in the same way that it would respond to a trisomy monosomy state that indicates some kind of issue. And so a lot of these cells die or are sacrificed very quickly and typically don't go on to cause any problems. And when it comes to if this cell is used, if this process happens during meiosis and makes a gamete cell that has one of these changes in it, and that gets chosen for fertilization and it forms a zygote, we sort of see the same thing that typically happens with most monosomies and trisomies, is that cell's not going to live very long, and typically there's no birth happening at all because the cells are removed or destroyed, or aborted in this case, fairly quickly. There was a story that I read that talked about how many falls, not really falls is the best word, how many, what's the best word here? Misstart's not the best word. Like how many times does fertilization occur but not progress past the 3-month mark? Usually that's sort of the cutoff for knowing you're pregnant is 2 or 3 months. I mean, nobody like data on, I forget what they said, but we probably shouldn't say anything like, because I'm just making shit up now. But it's more than one, I guess is the answer. A lot of people will experience a fertilization step and that cell mass is going to be gone before they even know that it was there. And it's typically because of the cell's natural reaction to an improper DNA content. So a lot of times maybe you're sort of late, and you're not late. It's because of some type of DNA problem in the zygote. But I don't want to be a downer on a Wednesday. It's nice on Sunday outside, so let's talk about fun things. Like what happens when you lose DNA in a deletion, or gain DNA in a duplication, or change the order of the genes in an inversion. Deletion sort of falls in the same category as monosomy because you're losing some big chunk of DNA. Duplication would fall in the category of a trisomy because you're gaining some extra DNA, which also affects gene dosage. But why is inversion bad? Who can think about a reason why changing the gene order is so bad? While you think about that, I'll draw a picture. I should let the screen down so you can see me draw it. I can see you all. I know what you're doing. You're laughing at me. That's okay. It'll be worth it in a second. Imagine if I taught lecture like this all the time. Would it be better? Would it be worse?
Be interesting.
It would be interesting. Anything about why inversion could be so bad? showing now the Wizard of Oz.
Could it be like amino acid order, like they're not created, like when you do gene expression, they're not created in the right order?
That would only matter if you took the gene and swapped it in between the promoter and the terminator, will it actually happen? But if you take the right amount of DNA, then the promoter and the terminator get swapped at the same time as the genes. Now for some things that will be an issue, and that is a possibility, yes. If you swap any amount of DNA and reverse it, now the codons or whatever are different order. Introns, exons can now be in a different order also, depending on what type of change you have. And so you basically hit the nail on the head. Everything's still there, but it's in a different orientation. Now, you might look at this and think that this is a weird, funny drawing, but what's wrong about one of these drawings?
Heart is upside down.
His heart is upside down, which means, where is his asshole?
His mouth.
It's right there. That's why it's sad. Because if you change the entire orientation of the body, now your mouth is down here and your butthole's up here. And that's the problem? Is it a problem? There's a genetic issue where your symmetry is reversed. And you know what it's called? I don't know what it's called, I forget. It's like the heart is on the wrong side and everything's sort of backwards. That creates some interesting problems. Everything's still sort of in the same general place, but everything's kind of backwards. And so inversion is bad because you don't lose information, you change the context of where that information is. Also in the context of the nucleus, DNA pieces are in certain locations based on how often we need to use them. If they get moved to the wrong place, maybe now they're more than you need. So who knows? I'll end up something that has a nice surprise. It's one of the weird things about chromosomal changes, how you actually get an inversion. Typically, it does require 2 crossing over events for that to occur. You have to cut the DNA in 2 places. For deletion and duplication, you cut once and you either take something out or put something in. It's a pretty easy thing to think about. The first 3, deletion, duplication, and inversion, are only affecting a single numbered chromosome. Because nothing is moving from chromosome 1, it's still chromosome 1. You're just losing something, or gaining something, or changing the order of something. It's also only affecting 1 chromosome, which is different than saying affecting 1 chromosome, or chromosome 1. 1 chromosome versus chromosome 1. Crossing over is rare, and that typically means that when you have a deletion, duplication, and inversion event, either the maternal chromosome is affected or the paternal chromosome is affected, typically not both. So you do create a real big imbalance in the DNA content and gene dosage by doing this thing. If it affected both paternal and maternal, maybe it wouldn't be that bad, who knows? Translocation is the only one of these 4 that will affect 2 numbered chromosomes. And so the idea of translocation is moving something from one position to a completely new position. Sometimes this can be what we call unidirectional, other times it will be what we call reciprocal. And so if you take something from chromosome 1 and slap it on the end of chromosome 3, chromosome 1 now gets shorter and chromosome 3 now gets longer. That's just a simple translocation event. A lot of times we actually see translocations as reciprocal. So when you cut chromosome 1 and cut chromosome 3, those 2 pieces actually swap positions completely. And you don't gain or lose anything. Still, the question is important to think about. You don't lose any information, you don't gain any information. But translocation is a bad result. And it's bad for the same reasons that inversion is bad. You change the context of where things are. So if you imagine you have a euchromatin region on the end of another chromosome, you've got euchromatin telomere, euchromatin, no more telomere, or maybe an extra telomere, who knows? And it's going to create a little bit of an issue there. So it's important to be able to understand what these things are doing, but also again, what types of chromosomes or how many chromosomes are affected by all these different types of things. And this is just a summary of some of those things that I was talking about. So maybe some go over everything again here. I'm not sure you can read it. The typical way in which I test your ability to understand these events is to give you a word. Like this word, which means nothing, but it is a word. It's a word that only has one letter used each time. And with this one word, we can see if something is changing. And so once you have the starting out word, we look at a change to that word. And you should be able to look at words and determine what is going on with each of these different things. So deletion, here we lost the J gene. We can just easily lose C and K instead of J, or U, B, and L. Deletions can happen anywhere, not just in the middle of a chromosome. Same thing for duplications. They can happen in the middle, or they can happen on one end or the other. It's just a random process where these things are going to occur. Sometimes more DNA is duplicated, sometimes less. Same thing is true for deletion and insertion. But what we need to look for is the loss of a letter for indicating a deletion event or a duplication of the same letters. That's an important distinction for duplication. We're not getting new letters, we're just getting more letters of the letters we already have, because we're duplicating something that was already present. And the idea that I want you to think about whenever we see new letters come into play is going to indicate a translocation event. We see here, we get formed letters that are not in the previous word at all, indicating that we got something from a different chromosome entirely. And here this probably represents a reciprocal translocation event because the Jack went somewhere else. But if we just added a snow to the end called Blue Jack Snow, that would still be an example of a translocation event. We're not getting X letters, we're getting new letters. And a lot of people struggle with the idea of deletion versus translocation, especially when the deletion happens at the end of a chromosome. And so it's a very simple idea to remember. Translocation is always going to introduce something new. If you don't see the introduction of new content, then by default the answer has to be deletion. And this is the format that you will always see on the quiz this weekend and on the exam in a few weeks. Here's what the original gene content gene order looks like. Something happens. Which new arrangement of letters matches that change? Or here's what they look like first, here's what they look like next. What happened to make that particular change happen? Make sure you can look at these things in both directions. Does anyone have any general questions about how you might go about identifying a duplication, deletion, inversion, or translocation event? Okay then. Crossing over. Genetic variation within the same numbered chromosome or affecting traits encoded on a single chromosome. Our second genetic diversity event is what we call independent alignment. Some textbooks will use a different phrase. I hate that other phrase because it's the same wording that Gregor Mendel used. Gregor Mendel used the phrase independent assortment. And because crossing over is tied into independent assortment, we can't use independent assortment as the name for independent alignment. Because independent alignment is part of the process of independent assortment. And this is just for me, you can use whatever you want in other classes, but for me, I'm going to be a real dick about it. And I like my language a certain way. So as we think about these things, please think about the way I want you to think about them, the correct way. That's right, the correct way. Always the joke. If you're not going to talk, then I'm not going to talk either. How about that? I'll just come in here and flip through the slides every minute. Seems like that. I don't like it very much, really. Maybe you like it more. Hard to tell. Because you won't tell me anything. So So how do I know?
I would have liked it more.
Thank you, at least one person cares. Everybody else can go suck a pumpkin, I guess. Did you paint your pumpkin or did you still? I don't see any paint on it. What does it look like? Well, what I want you to think about is when Gregor Mendel was talking about independent assortment, he had no idea about chromosomes or genes or whatever. He just knew that the things he was looking at, the traits, were acting independent of each other. Now, based off of the analysis of the pea plant, we know that several of the traits are on the same chromosome. So the only way to deal with the separate inheritance of traits on the same chromosome is in the context of crossing over, breaking those things apart from each other. And so both of these things are what Gregor Mendel was observing when he put together his idea of independent assortment. Which is why I really like you to use this name as for what happens during metaphase 1. And again, with crossing over is just what we always have for prophase 1. So maybe a long-winded explanation of why I like the name independent alignment more than independent assortment. Also, alignment sort of more fits the idea thematically of what metaphase does. Metaphase moves the chromosomes to the middle. They line up at the middle. They don't assort in the middle. They line up in the middle. I think of anaphase as the assorting part, because I think of sorting as pulling things apart and separating things. So independent alignment basically says that chromosome 1 will orient itself relative or independently relative to all the other chromosomes that are present inside the cell. Chromosome 1 is not an independent unit. It is 4 pieces of DNA stuck together in what we call a tetrad. And so it's better and more appropriate to state that the tetrads line up at the cell midpoint independently and randomly compared to what the other things are doing. And because of this randomness or independentness, the alleles can be on opposite sides of the cell. And just in a basic setup where we have this heterozygous setup, this represents what we call one possible metaphase 1 alignment. Because we can see that all the capital letters on one side, all the lowercase letters are on the other. But this is only showing you one of the possibilities. And if we sort of think about this, these are the other possibilities that would exist. Why are there only 4? Why aren't there actually more? And what I want you to think about is, what if it lined up like this? What we see on the board is the opposite. But why aren't both these listed?
It's basically the same because the same things are paired together in different
Yeah,
cells.
so even though it's a mirror image, it might look different. It's not different because of the symmetry of what we want to think about. Anything on one side is going to go together. So it doesn't matter if it's on the left side or the right side, it's that they're on the same side together. It doesn't matter left or right. Left and right really have no meaning. It could be up and down for all we care. This thing could be diagonal. But it's what side are you on? It's actually, what are you with more than what side are you on? And so as we look at going from sort of all homozygous, so all dominant one side, all recessive on the other, we just change one each time to get all the different variety. What if we change 2 of them? What do we get if we change 2 things? Let's change A and B. Instead of just A or B, what if we change A and B? What does that look like? Why is that represented here? Why is it the last one? A and B are on the same side. This A and this B are on the same side as that A and B. So how did you split them? I think you're on the right track, yes, but you're sort of getting what I'm trying to get to is, a lot of people look at this and think there should be more possibilities because you switch 1, then you switch 2. And that change might change the order, but it doesn't change what is on each side. So it might look like you changed E here, you just split the E, But you could also flip the A and the B and you give the same orientation. So you need to sort of worry about. Sometimes not worry about as many possibilities. There is a way to calculate the total number of possibilities, but nobody loves math, so we won't talk about how you do that, although it's on the next slide. But what is going to create genetic diversity? Homozygosity or heterozygosity? Okay, it's the same answer that we had for crossing over. It's only when there's differences between maternal chromosome and paternal chromosome that lining up orientation matters. So in this case, there's 3 heterozygotes. That makes the 4 different possibilities. But if you ever throw in a homozygous state, you don't increase the number of possibilities because you're going to get capital A on one. In this case, capital A is on both sides, regardless of how you flip things. And typically, I don't ask you to identify these things because it's kind of a pain in the ass. Well, let's do a test. Humans have 23 pairs of chromosomes. If you have individuals that's heterozygous for your maternal and your paternal chromosome, how many different alignment possibilities are there? What is n? It's 23. Yeah, so now this is n to the power of 22. So what's 2 to the power of 22? I'll get you started. 2 is the power of 1. 4 is the power of 2. 8 is the power of 3. What's next? 16. What's next? Let's keep going. How many possibilities are there? Just from doing independent alignment? Like 6 million something or other? I forget exactly what the number is. But just from aligning the DNA in different ways, you get a lot of different possible combinations of things. Sometimes you can get half maternal on one side, half paternal. It could be even, 50-50. We ignore the sex chromosomes, just the autosomes. It could be 1 to 21. It could be 2 to 20. You never know what possibilities you have. And if it's only 2 maternal, which 2 maternal is it that you get? So there's a lot of genetic diversity just inherent in the tetrads moving to the middle in a different orientation relative to everything else. When a cell undergoes meiosis, how many of these possibilities does it actually encounter? Does one cell make all 4 of these? Or does one cell only make one possibility? This is an easy question.
Can you ask the question again?
No. I'll just answer it for myself. The question was, when one cell starts meiosis, does it make all 4 of these, or just one of them? Right, it's one cell. It can't make all of the possibilities. You can only make one. So even though there are 6 million possible combinations of aligning the chromosomes in humans, only one of them will actually occur every round of meiosis. So at any particular point in your meiosis cycle, you never know what you're going to get. I love Forrest Gump for this reason. You never know what you're going to get. There's a lot of variety out there. And throw that variety on to the fact that crossing over is going to happen for every chromosome. You don't know where the crossing over is going to happen for the chromosome, it's going to happen once. Extra diversity you can't really account for or have an equation for. There's a lot of genetic diversity just inherent in these 2 things, producing these gametes. But we also have to keep in mind that the last bit of genetic diversity comes in from your other parent. What DNA are you getting from your other parent? And how much diversity went into making that gamete also? There's a video of Neil deGrasse Tyson. Does anybody know Neil deGrasse Tyson? He's an astrophysicist. He was talking about the genetic diversity in humans. And I think he said like there were 9 trillion or 9 billion, it doesn't matter. If you were to take all the genes in humans and sort of shuffle them up to make different varieties, you get like 9 billion, I think, 9 billion? Maybe 9 trillion. Maybe it's 90. Makes it doesn't matter. His basic point was that there are more genetic possibilities than there are humans, or will ever be humans. So the people we see, the genetic diversity we see, is a small fraction of all the possible outcomes that are out there. What do you guys say? Life's cool. But during prophase, we do crossing over to create some genetic diversity. When our tetrads form, then our tetrads line up at the middle of the cell, independently of each other to create a little bit more genetic diversity. Then we move to the next phase, which is to separate those things. And so after they line up, there's no more genetic diversity introduced during the process of meiosis. So we pull things to separate sides and continue the rest of the process. Remember, the tetrad is lining up, so the homologous chromosomes are also lining up. And when we do anaphase, we break the tetrad and pull the homologous chromosomes apart. Each chromosome is still a sister chromatin pair, which is why we represent it in this fashion. So we can start with 4 pieces of DNA in the middle and pull that apart, so we get 2 going to either side. And we pull them for a certain amount of time until we are ready to move on to the next phase, which is telophase. And remember, during telophase, we're going to reform the nucleus. Chromosomes can decondense, and all the microtubules and kinetochores can be removed and destroyed and recycled. And then cytokinesis occurs, and we move on to the next phase, which is prophase 2. Remember, prophase 2, we don't see any synapsis. There's no longer homologous chromosome anymore. There's also no point for crossing over to happen. So it goes directly from prophase 2 into metaphase 2. And so for metaphase 1, the tetrads will line up at the center, and because there are tetrads or homologous chromosomes, there's going to be a variety that exists because of how they line up in the center. But when we talk about meiosis 2, we always want to think about there is no genetic diversity or variation possible, because we're dealing with DNA that should be completely identical to each other. Now the question is it really? Are the 2 chromatids of a sister chromatin pair completely identical to each other at this point? Right, the answer is no if we assume that every chromosome has one crossing over event. Chromatids are not completely identical to each other. We still don't talk about there being an independent alignment phase here, but it could matter how these things would line up, because one might be all dad, the other might be all mom, but some might be slight variations of those combinations. We still don't talk about there being an independent alignment phase during metaphase 2. We kind of glossed over the fact of crossing over and just think about the sister chromatids are sister chromatids, therefore they should be completely identical to each other. Therefore, it doesn't matter how they line up, because even Stevens separation is going to happen. We should appreciate that they're not going to be exactly the same, so there's going to be a little bit more variety that can occur at this point. In the same machinery, the kinetochores and microtubules that are used to move the DNA to the cell midpoint during metaphase 1 are also in play here to move the DNA to the middle. Once it gets to the middle, it is going to move apart. So our sister chromatin pairs line up during metaphase 2, and the sister chromatids will be separated during the anaphase 2 stage. Now if they're apart, we can call them individual chromosomes again. We don't refer to them as being chromatids anymore, because chromatids is typically a term designed to talk about the 2 pieces of DNA that are always stuck together. The 2 pieces of DNA that arose from a duplication event. Not to be confused with a chromosomal duplication change. It's kind of unfortunate we use the same words a lot of times to mean different things. We copy our DNA at the very beginning of the process, and a chromosome becomes 2 chromatids, and then here at the end of meiosis, we pull apart those sister chromatids to make them individual chromosomes again. And telophase 2 happens, which again, we get rid of the kinetochores and the microtubules, the chromosomes decondense, the nucleus reforms, and the cell sits there and waits for something to happen. Nothing happens, the cell gets recycled or discarded. A lot of people describe meiosis II as analogous to mitosis. This might not make any sense right now because we haven't talked about mitosis, but it will when we get to mitosis, so it'll make much more sense. But meiosis II is pretty straightforward, not a whole lot of variety, not a whole lot of change going on. When it comes to meiosis I, we are doing two really important things: synapsis and crossing over during prophase I, and then independent alignment of tetrads during metaphase I. You can always want to remember the names of the DNA molecules that are separated during each meiosis phase, homologous chromosomes being separated during anaphase I, sister chromatids being separated during anaphase II. Does anybody have any general questions about meiosis so far? We understand crossing over, we understand independent alignment. We have a basic understanding of metaphase is the lineup phase, anaphase is the separate phase. Different things line up during metaphase I, then metaphase II, different things get separated during anaphase I versus anaphase II. There is one more possible problem that can occur during meiosis. The first is, again, those chromosomal abnormalities caused by crossing over going wonky. The other is for the separation of DNA to not occur properly. This is a process we call non-disjunction. Non-disjunction is the ultimate double negative, because non-dis. That is called the junction, because that's what really happens. Things stay juncted. What is a junction? A junction is a connection, and a disjunction is a separation, and a non-disjunction is no separation. So you know why this term came about. It's very literal. The scientist that first observed this, his name is Bridges. He's working in the same lab as Thomas Hunt Morgan, which we'll talk about a little bit about Morgan later on. But they first noticed this when looking at the inheritance of the sex chromosomes in flies. And noticed that some flies didn't have the same sex chromosome content as other flies. He was curious why, and so they studied the process and figured out that non-disjunction is happening, where the chromosomes or chromatids don't separate properly. Like crossing over, non-disjunction is a rare event, but it's much more rare than crossing over. Because crossing over is always going to happen, but non-disjunction is very rarely going to happen. But it does have two opportunities. It could happen during the first separation stage, anaphase I, or it could happen during the second separation stage, anaphase II. So it's really important to remember, anaphase is where we're separating DNA, so non-disjunction is going to happen because of a problem with anaphase. The chromosomes don't separate properly. Non-disjunction is more common during egg production or oogenesis than it is in spermatogenesis. There's a fun little story, I don't know if you know, that for women, all of your eggs are formed before you're even born. They're frozen in time in a meiosis somewhere. It doesn't really matter where, because we're not talking about that. But meiosis has started, but paused for every single egg a woman has. So as you get older, things sort of stop working as well. So a natural thing, things sort of break as they get older, don't work as well. So every time a woman ovulates, an egg has to be, meiosis has to be finished. So we go through the separation of DNA and all that jazz, which is why the separation becomes more of an issue as you get older, because the machinery to do it is not working as well as you get older. We'll talk about a problem that is going to happen for men and sperm production, but it's not non-disjunction, it's on something else. But non-disjunction is the thing that happens that leads to monosomy and trisomy events. But monosomy and trisomy only refer to a zygote, so after the fertilization step. Meiosis does not create the monosomy or the trisomy event. The fertilization process generates the monosomy or the trisomy event. And so as we think about the possibilities, we can have non-disjunction during meiosis 1, and we can have non-disjunction during meiosis 2, but when it happens at the very beginning, we will not get any normal gametes. That's something that I would like you to remember. No normal gametes are produced when non-disjunction happens during meiosis 1. So we fail to separate our homologous chromosomes, meaning both the maternal and the paternal chromosome 1 go to one cell, and the other cell gets 0 of chromosome 1. And that's the problem there, is you lacking a complete chromosome that you need. Whereas the other cells normally get 2 copies of chromosome 1, maternal 1, and paternal 1, for example. Now on the other hand, if non-disjunction happens during meiosis 2, we have already successfully separated our homologous chromosomes from each other. So we're going to get some normal gametes. Basically we'll get 2, because half of the cells are going to be okay. The other half will be bad. Again, the bad is always either too much or too few. You're either missing one whole chromosome, or you have 2 copies of one whole chromosome. And so instead of having 1 or 3, you have 0 or 2. So the 0, when it fuses to another gamete with 1, creates the monosomy state. When a 2 fuses with the normal gamete, you get the trisomy state of 3. There's a slide in here somewhere that talks about non-disjunction. I think it's at the end. Yeah. So at the end of all this, I put together some cartoons for you to look at. This will illustrate some of the things of meiosis. We do have just one more little topic to talk about, and we have plenty of time to talk about it in. So yay! I'm going to talk until 1:51 today, according to my clock. But don't hurry, I won't make it. But one of the problems that scientists sort of had to deal with early on, which is why Bridges and Morgan were doing the work they were doing, is do the autosomes and the sex chromosomes do the same thing? And one of the first questions was, in males with an X chromosome and Y chromosome, how do those work? Most people sort of understood that an X and an X can go together easily during synapsis because they're the same DNA. But for a long time, X and Y, like why do X and Y go together? It wasn't until we had the DNA sequence analysis to see those pseudo-autosomal regions that we knew that X and Y would synapse together. There's even crossing over that happens between the X chromosome and the Y chromosome. But it's only within those pseudo-autosomal regions. Again, it's not anything you need to think about, but X chromosomes and sex chromosomes were originally identified in insects. So that's yay, bugs. Dr. Pitts will be happy. You can tell him I said so. We talked about this already. I just mentioned a few minutes ago. X and Y do have some similar DNA sequences. But the real point of going through the slides is to mention another scientist, and his name is Thomas Hunt Morgan. And while Mendel might be the sort of the first father of genetics, Morgan might be the second father of genetics because of his important work studying the inheritance of chromosomes, particularly showing that there are things on the X chromosome that are not important for sex, but are important for other things. And for Morgan, he was studying eye color and a few other things that are encoded on the X chromosome. So led to a new line of studies that sex chromosomes do matter. I mean, we need to worry about their inheritance, just like we need to worry about the inheritance of the autosomal chromosomes. But they act just like a regular chromosome. They come together during synapsis. Crossing over can happen. Crossing over between X chromosomes, will that create something new? or not make something new? Are your X chromosomes exactly the same? Right, they're not, right? So your mom's X chromosome, or one X chromosome from mom, one X chromosome from dad, not gonna be completely identical to each other. So crossing over could make some genetic diversity between one X chromosome and another. And then we inactivate one of those chromosomes. So now which chromosome gets inactivated also going to affect some of your cells in your body differently than some of your other cells in your body. And if you have questions about how meiosis works, you can look at the last set of slides, and it'll also show you a little cartoon of what happens if non-destruction happens. I'm not going to show it to you right now, because they're there for you to look at outside of class. Okay, you talked me into it. I'll keep you here longer to show you these slides. Now make sure you can draw these slides, because I will ask you to replicate these slides with 100% accuracy on exam number 2. If you thought 75 questions in 50 minutes was hard, this will take longer. Nobody likes that joke. No, not everybody gets that joke, because not everybody has to sell logic. For those of you who do, I think it's funny. I'll darn you. Or full of me joke, right? Hopefully you like it. Hopefully you like it. But in these last 2 minutes of class, I'm going to start the next set of slides, which I haven't posted yet. This set right here. And if you didn't like the math of exam 1, here's where some really fun math gets to show up. It's not as bad as Hardy-Weinberg, but it is on the same level as Hardy-Weinberg, if you know what that is. So yay! Bring your money on Friday, we're gonna play some blackjack. And I will tell you the house always wins, regardless of what your cards are. So just bring in money on Friday. It'll be good. Because after this class, hopefully you can go to Vegas and make some money, because you know the probabilities, you know when to hit and not to hit. As the gambler says, know when to fold them, know when to hold them, know when to stay, and know way to run. That was your cue to know when to stay and know when to run. If you don't know the game,